Can you guys? All right. Okay, we're going to reconvene and get started in a minute. All right, thanks for your patience while we sort out our Zoom. I think we're good to go now. Our next two presenters are from the Jefferson Project. Dr. Allison Hrycik is a research scientist for the Jefferson Project, and she's based locally to research harmful algal blooms on Chautauqua Lake. She's originally from Colden, New York, and holds a Ph.D. in biology from the University of Vermont. During that Ph.D., she studied how changing winter conditions help set the stage for algal blooms during the open water season. Her current projects include monitoring lake bottom algae and understanding nutrient inputs in the Chautauqua Lake watershed. Joining Allison today is Dr. Harry Kolar. He's an IBM Fellow with IBM Research, focused on environmental monitoring and management. Dr. Kolar currently leads a multidisciplinary team of IBM researchers in the environmental sciences domain and is the associate director of the Jefferson Project. Dr. Kolar holds BS and MS degrees in physics and an interdisciplinary PhD in the science and engineering of materials. And today they're going to give us an update on harmful algal blooms and water quality in Chautauqua Lake.
Are we set, Toby?
Yep. So Harry's going to start off the presentation, and then I'll come in a few slides.
Okay, let me know when you're ready.
We're good to go.
Okay, great. Well, good morning, everyone. I'm sorry I couldn't be there in person. I would much rather be there. But I'm really pleased to have a time slot on the agenda for the Lake Conference this year again. So thank you very much. And it's a very special year with the 150th anniversary. And we're very happy to give you some updates here. I'm going to start off and just basically do a quick refresh. Many of you are familiar with the Jefferson Project, but in case some of you aren't, I just want to hit a few points, sort of draw out the big picture, and talk about really the long-range plan of what the Jefferson Project means. While harmful algal blooms are one of the critical issues, obviously, at the lake that we're working on, and that's why we have specialists, scientists like Allison actually there to work on it, which we're very fortunate to have. There are other things to be concerned about for the lake's long-term viability, safety, health, et cetera. So it's all about preservation and sustainability. And I think from that standpoint, it's very, very important. So let's go to the first chart. I'm going to just hit a few up front, and then most of the balance of the presentation will be Allison talking about the actual results. So again, Jefferson Project started, basically it's in its 11th year now. It originally started with RPI and the Fund for Lake George, which is now the Lake George Association. They merged in 2013 at Lake George. So we've been 10 years. There's actually a 10-year pretty nice glossy report on that project you can find on the Lake George Association website if you want to see the types of things we were doing. It covers a wide range of things. We moved to Skinny Atlas in 2018 based on a harmful algal bloom there that occurred, the first one ever that was quote unquote documented and official in 2017. Skinny Atlas was also at that time an oligotrophic lake, which means very, very low nutrient levels, very clean lake, also used as a water supply for Syracuse, so very different from, let's say, Lake George, but very, very similar water quality. And it was just a place that harmful algal blooms shouldn't occur. And then, of course, subsequently, we've had the same problem at Lake George. Chautauqua, we came in 2021 and started with a pilot project, and we built on it from there. So we've got a team of scientists with diverse specialties, basically understanding water quality issues, especially things like anthropogenic human impacts, if you will. And it's a very, very important project from that standpoint. Obviously, the lake is a vital resource to the region, to the institution. It's absolutely critical. And a lot of things we're building are working towards building into programs at the institution for lake preservation and leadership in these areas, including things like policy, links, et cetera. So next slide, please, Allison. So you're going to see this slide, and you've probably seen it before if you've come to any of these presentations, but it's a pretty standard slide, and Allison will hit on some of the points when she gets into her presentation, but I just want to set it up. This is kind of the big picture of what goes on at the lake. There's a lot going on. It's not just a sort of quiet body of water. It has multiple layers in it in terms of the density of the water having to do with temperature. There's dynamics involved with it. A lot of it's driven by weather. So wind is very, very important, et cetera, temperature. You've got runoff effects, and then you've got a whole series of things like thermal setups vertically through the lake, understanding what the water currents are doing. There are surface currents. There are internal waves that actually build up in the lake, which means there's movement of water, if you will, at lower depths, all the way down to the bottom in many, many cases. And there's also transport mechanisms for waves to bring material up from the bottom of the lake. And then there's a whole interaction of how all this ties together with things like the bottom, the sediment, runoff, any impurities, if you will, nutrients, et cetera. So it's really a combination of a lot of things. Not all of them are actually here, but I mean, interaction between physics, biology, et cetera, is extremely complicated. So we have a team of people, limnologists, lake specialists as well, that are studying this. And we use a combination of different approaches. If we can go to the next chart, please. Based on state-of-the-art technologies and methods, there are things that the Jefferson Project has developed over the past 11 years that are being utilized and that Chautauqua Lake has been benefiting from. You've all seen, probably you've seen the vertical profilers on the lake the past couple of years. They've actually changed in color, if you will, because the new models have actually been deployed. The model you see in the upper left-hand corner, these are basically the robotic platforms that measure what's going on from top to bottom in the water column, and they're out moored in the north and south basins. We do a lot of different types of samplings, but now this year, what's different, and Allison will talk a little bit about that as well, is the new tributary monitoring station. So this is the first wave of stations to understand and to measure what's coming in from the major tributaries and, let's say, catchments or basins, if you will, and how they flow into the lake. So it gives us all kinds of information on water quality, flow, et cetera, and we're now better able to characterize and understand what's happening. In the middle, there's a bunch of things. Also, you've seen the model in the center here, which is basically the transport model. We get into things like hydrodynamics to understand how the water is moving within the lake. All of this ties into a series of models, which then allow us to eventually build a model to understand how the nutrients flow, which are a very, very key point of the project. I will also make one comment here is that we're very, very grateful for local support. The stream stations, the first 6, there's going to be probably another 6 the next stage of the project with additional weather stations as well that will be rolled out. So there's going to be additional infrastructure coming out. There's another important piece of it that, I mean, these are locally built sheds. These house all the sensor components, if you will, electronics tie into the network, and then there's some pipes leading from the tributary monitoring stations down to sample water. We can actually extract samples, et cetera, mechanically timed them with computer code to make decisions on when to sample things. But we're very grateful for support, local support for landowners who've allowed us to put these on their land. They're also being built by a company called Stantec, which is one of our partners in the project. They're special design that they're environmentally low impact. Normally these things, and if you go to Lake George, you'll find many of them are poured concrete slabs. Certain areas of the ground has to be disrupted, if you will, sort of more permanent structures, et cetera. These have these wonderful sort of helical screw mounts. They basically have these large frames that have these helical screws in their standoffs that are basically wound into the ground. They look like big screws, literally, and they sit on top of that. So they can be moved, they can be repositioned, and they're low bit impact because the ground doesn't have to be prepared. So this is kind of a first. That design was the first one made for this kind of a system here at Chautauqua Lake. And similarly, we've had a lot of support, local support from people on like the algal tile surveys. So we thank you for that. Next chart, Allison. So very quickly, this is a big picture. Again, you've probably seen this, but this is what brings it all together. We have on the bottom, we have a series of different types of sensors. We have floating sensors on buoys, as you will see. We have things like weather stations on terrestrial weather stations, which we hope to have, I think, 6 out next year. We also have the stream stations, and we also have other measurement devices that look at like water currents. So while we also use that information flows into an analytics platform, so we collect all the data, it's all standardized. The benefit of that also is that we use standard data formats that can be shared, so we can work with other scientists, which we do in a collaboration mode. But we're also using all the same for Lake George, for any other lake that we work at. So the data that's already been collected, which is massive at Lake George, both modeling data and observational data, meaning sensor data, is being stored, and we can look across all those. Even though the lakes are different, there are similarities, so we can look for patterns to better understand things, to better look at mechanisms that are involved, et cetera. And then we couple that with computer models, which is that stack in the right-hand side that basically does things like weather, land surface, where the water is going, what the stream network looks like, what the soil interaction looks like, then how both of those combine into the circulation model, what's going on within the lake, and then looking at things like the chemistry and the biology of the lake. So all of it's tied together. Eventually, and on the biology side, we're building things like early warning systems for HABs, which we're getting started on and have made some pretty good, significant breakthroughs, again, which is absolutely important because of the time it takes for identification and laboratory analysis, et cetera. So we're getting a big picture. So all of this is very dynamic. This is not just you run it once and see what's going on and examine it, et cetera. This is all functional. We're collecting data right now from the new stream stations, et cetera. So it's a big investment, and we realize that, but it's building a data corpus, a body of data, which is going to be useful from here on. It's establishing a baseline, and we're filling in a lot of the holes of things that we don't know at this point in time. The more data we have as far as from a time standpoint, over periods of time, over the seasons, and also the density of that data, as well as what it looks like geospatially. In other words, how frequently are you sampling in certain areas, how close together, et cetera, how much coverage you're getting on the lake is all very, very important. So I wanted to set that up. The point being that the investment and the progress that's being made is going to last for a long time, and it's going to basically present and hold Lake Chautauqua Lake in the mode of having a lake that is very, very well managed, because all of these things are tried, tested, validated in terms of models, et cetera, and it can prove that you're making differences, because you're going to be able to make sort of what if kinds of calculations of what if you would do this, what if you reduce the nutrients here or there, how you do that, different story. But the point is then you start making these measurements, and it's the old story, if you can't measure it, you can't manage it. And that's very important, number one, to make sure you're doing the right thing scientifically and environmentally, but also the right thing when it comes to looking for funding, because you can basically prove your efficacy. So I'm going to leave it right there, but that's the big picture. There's a lot to it. Time, as we looking at the next phase of the project, will hopefully sort of build out almost all the infrastructure. So the network is going to be very, very dense. And we're going to have a lot of sensors in the water, more tributary stations, weather stations, which are new, as I had mentioned, et cetera. So I'm going to go ahead and turn it over to Allison, and she's going to take you into results to bring you up to date from last year. Thank you.
All right, thanks, Harry. All right, so before I get started and some of the results that we found over the past couple years, I just wanted to mention, stand closer to the mic. Thank you. I just wanted to mention that we do have a report online from all of our field work last year and some data from previous years. That report is available on the Chautauqua County website. So I'm going to be giving a quick overview of what we found, but if you're interested in reading more, ask me for the link. I'm happy to send it to people, but you can find it on the Chautauqua County website. All right, so getting into some of our research on harmful algal blooms. So harmful algal blooms, I might call them HABs sometimes. This means different things to different people, but on freshwater lakes like Chautauqua Lake, this typically means that there's an overgrowth of cyanobacteria. And if you hear blue-green algae, that's the same as cyanobacteria. Those two terms are interchangeable. So these blooms can be harmful to humans, pets, and wildlife. So that's why we see these warning signs on beaches to not recreate, to not let your pets in the water if there's a HAB. And it's because they contain toxins, including neurotoxins, which go after the brain and nervous system, and hepatotoxins, which affect the liver. So you should be cautious around these HABs. And it's important to note that not all blooms that you see are toxic, but it's impossible to tell which ones are just by looking at them. You have to actually get a lab test done to see if there are toxins in the algae. And Chautauqua Lake is a really interesting place to study HABs because, as Courtney had mentioned, the north and south basins are very different from one another, but there is some exchange between the two basins. And there's this transition zone in the middle of the lake. But in general, the north basin is pretty deep, it's a lot colder, and it has less frequent algal blooms, whereas the south basin is shallow and warm and tends to have a lot more algae. And so the ways that we study this, Harry has hit on some of this already, but I'm just going to get a little bit more in depth. One of the ways that we measure water quality parameters, including HABs and their drivers, is to use these vertical profilers. You're probably familiar with these if you've been boating on the lake. They look like these very large orange pontoon boats with solar panels on them. But what they do is they have a lot of sensors that are measuring different water quality parameters. Some of the sensors are attached to the bottom of the platform and measuring right near the surface, and some sensors are constantly moving up and down in the water column. So these sensors are measuring things around the clock. They're constantly collecting data. This includes things like temperature, oxygen, chlorophyll or BGA, which are both measures of how much algae is there. They're measuring water currents, water clarity, and then this post that's sticking up on top of the profiler, that's collecting a bunch of weather variables, such as solar radiation, wind speed, and wind direction. So we have two of these profilers on Chautauqua Lake. We usually put one in the North Basin and one in the South Basin. In addition, we have a physical lake sampling program. So this is when we go out on the boat and collect a bunch of samples to measure water quality parameters. This includes things like nutrients, algae, zooplankton, and water clarity. This program changes a little bit every year as we adapt to our research goals that year. But for example, last year we sampled every other week at 18 lake stations. And in previous years, we had also measured toxins and done some genomics work to see what cyanobacteria species were there. So not only does this give us a lot of good data, it helps us ground truth and validate what we're seeing from those sensors and to expand what we learned from those sensors to other parts of the lake. So put together, we have sensor data that is running around the clock, constantly collecting data, and then we're able to ground truth that data and also see what's happening in other parts of the lake using this physical lake sampling program. And this is a very intensive monitoring program. Last year alone, we collected over 1,100 samples. So to get into some results of what we've seen so far, this map that I'm showing is what we found for one species of cyanobacteria called Phanozomenon, which is a toxic species. These are data from August, and the darker spots show where there was more of that cyanobacteria. You'll notice there was quite a bit around the center of the lake, near Long Point and Bemis Point, but also quite a bit in the South Basin. That's kind of what you'd expect for cyanobacteria, is that we find more in the South Basin than the North Basin. And that's just one species, but there are some other key players. Microcystis, which is shown on the top right over here, shows similar patterns, and it tends to show up kind of later in the season along with the Phanozomenon. This one on the bottom, called Gliotrichia, that's more of an early season cyanobacteria. That's probably what is in the lake right now. You can actually see that if you're looking at the water. It looks like tiny little pom-poms floating around. And Gliotrichia is interesting because in other lakes it can be toxic. It can release toxins, but here we've tested a lot of samples, and so far it does not seem to release toxins. We're not sure if that's just the genetic strain it is or if it's some environmental variables that are limiting it. But it tends to be gliotrichia early in the season and then switch to other stuff like aphanizomenon and microcystis later in the season. And Courtney and Harry have also introduced this idea of nutrients. So nutrients are what are helping this algae grow. And when we say nutrients, we're mostly talking about phosphorus and nitrogen. And so here I have some phosphorus data shown. We measure a lot of different forms of both phosphorus and nitrogen. And I have total phosphorus here on the left. This is through the season last year. And you have the North Basin with the black lines and the South Basin with the gray lines. Each line is a different one of those 18 sites that we sampled. And you'll notice with total phosphorus, we have higher phosphorus in the South Basin, but it's really variable. Everything's kind of all over the place. There's a lot of variation. When we look at soluble reactive phosphorus, which is a certain form of phosphorus that can be easily taken up by algae, so it's really good algae food. You might also hear this called bioavailable phosphorus, or it's measured as phosphate. So with this soluble reactive phosphorus, you see the opposite trend, where you have more in the North Basin and less in the South Basin. And this is actually just a signal of a lot of it being taken up by algae, because there was already a lot in the water column. The algae are using it up, using it to grow, and so it ends up depleted in the South Basin where there are a lot of algae. So I haven't shown nitrogen results here, but we can think about those sorts of things for different forms of nitrogen as well. And so when we're thinking about what drives HABs, the common thinking is that if we have a lot of these nutrients, nitrogen and phosphorus, and we have really warm temperatures and a lot of sunlight, then we end up with a surface HAB. However, it's not that simple. It's, of course, never that simple in lake science, and that's because there's a lot of different stuff going on at the same time that Harry had alluded to with that system of systems diagram where everything's interacting. And so one of the ways that we can break down the nutrient piece of this is to think about internal loading versus external loading. Internal loading is nutrients that come from within the lake, usually from the sediment, and external loading is nutrients that come in from outside the lake. So this is stuff that's coming in through streams, through overland runoff, through any discharge sources, maybe even from groundwater. So it's important to kind of parse out where all of those nutrients are coming from so that we can better direct management of where are the bulk of the nutrients coming from. So Courtney had talked about this a little bit with internal loading. So we have some more data on that. So internal loading for phosphorus is coming from the sediment. So what happens is that there's a chemical reaction that when oxygen is low, phosphorus is released from the sediment. And we're able to measure that in Chautauqua Lake. Here I'm showing the North Basin. The graph on the bottom is similar to the heat maps that Courtney was showing, but it's oxygen from July to November in this case. And so that blue blob on the bottom left is that time period of very low dissolved oxygen at the bottom of the lake in the North Basin. And that happens through the summer until about mid-September, or at least that's when it happened in 2022. The graph on the top shows soluble reactive phosphorus, so that's that bioavailable phosphorus. And you'll notice that the longer the oxygen stays low, the more of that phosphorus gets released. And so this is just showing the North Basin, but something similar happens in the South Basin as well, but it just happens for a couple days at a time. It's like these small pulses of phosphorus being released. So dissolved oxygen, just to reiterate what Courtney was saying, dissolved oxygen is really important to understand in Chautauqua Lake. It's really a key variable because it's really rapidly depleted when there's stratification. So in the North Basin, dissolved oxygen is at 0 at the bottom of the lake for a lot of the summer and possibly other times of year. And this layer of low dissolved oxygen can't really support any complex life. So for example, fish can't live at the bottom during that time for much of the season. And it's also an important indicator of internal loading of phosphorus. So oxygen is a really key variable to understand what's going on in the lake. So our current understanding at this point of HABs in Chautauqua Lake is that we have abundant nutrients here. We have lots of phosphorus, lots of nitrogen. However, the sources of all those nutrients are not that well understood. In the north and south basins do behave very differently from one another, but we do see some exchange between the north and south basins. So it's also important to understand what's going on in that central part of the lake where there's some exchange. And so working towards a better understanding of nutrient dynamics will really help inform management because we'll be able to zero in on where a lot of those nutrients are coming in. So our next steps are working towards a nutrient budget. You can think about this like balancing your checkbook where you have some stuff, you have stuff coming in, you have stuff going out, and you're left with the balance that's there. And so in the case of nutrients, you can do this with both phosphorus and nitrogen. You can add up the amount of nutrients coming in from external loading and internal loading, subtract the exports, so that's things like nutrients being taken out in the outflow or just being taken up by plants or algae, and then you're left with the nutrient concentrations. There was a previous nutrient budget written for phosphorus in 2012. However, it wasn't very high resolution, so we're hoping to build on that and get a lot more information to improve it. So to that end, that's where we've changed some of our research goals for this year. So for the vertical profilers, we'll be launching those a lot closer to the center of the lake this year so that we can get a better handle on that interbasin exchange. One of them will be just above Long Point, and the other one will be just below the bridge. And then, as Harry mentioned, we've started our tributary station rollout where we measure water quality and nutrients coming in from 6 main tributaries. Right now, these are on Mud Creek, DeWittville, Prendergast, Bemis, Ball, and Goose Creeks. But in the future, we hope to add 6 more stations and some weather stations. And these stations now are all built and they're starting to bring in data, which is great news. All right, so just to conclude, we know that Chautauqua Lake has persistent HABs, and that's linked to abundant nutrients for algal growth. However, there are a lot of factors that lead up to a HAB. So this nutrient loading especially has taken decades, maybe even centuries to get to the point where we're at, but some other factors like the weather conditions that help trigger a HAB, they only take a couple weeks of buildup. Maybe it's just one day that the conditions are perfect. But then all of this stuff culminates at once. And sometimes it's easier to understand these immediate triggers, like the right weather conditions, than it is to understand these long-term drivers that build up over time, like nutrient inputs. But overall, nutrient reduction is really key for reducing HABs. And the way to get there is to make a high resolution nutrient budget that will allow us to better pinpoint the most useful areas for management, because we'll be able to pinpoint which sources of nutrients are the most important in Chautauqua Lake. So this work has been done through a lot of collaborations, so I just wanted to acknowledge a lot of the organizations that we've worked with. Thank you especially to SUNY Fredonia for your collaborations and for hosting me as a visiting researcher, BGSU for research input, and then Chautauqua Institution, Chautauqua County, and the Sheldon Foundation for funding, as well as local partners like the Alliance, the CLP, and the CLA for your collaborations. And we're always open to more collaborations and interesting new research questions. So, talk to me if you're interested. And Harry is still online, so if you have any questions, feel free to ask either of us. Thank you.
All right, we got some questions rolling in. Let's see. The first question I had, and this sort of picks up on the temperature stuff that you were talking about, and Courtney was talking about earlier as well. Real simple question, just says global warming in the lake. They saw that it was 80 degrees off of DeWittville, And I think that really the question is, what impacts do you see as climate change continues with the research that you're focusing on?
Sure. Yeah, so that's kind of always in the back of our mind as lake scientists. How is climate change affecting the situation? So I'm not sure about specific temperature trends. Courtney might be able to answer that a little bit better, but one of the things that I've been thinking about a bit is this potential decrease in ice cover and what effects spring warming has. And that's something that we have some ice data, but we don't have a lot of data on this early spring season when we expect to see the most changes. And so that's something I'm very interested in researching in the future, and we have plans to do a little bit of that in the future, including the project, that thin ice project that Courtney was talking about. As far as summer conditions, there have been a lot of published studies lately that say that the combination of warmer temperatures and increased nutrients have increased harmful algal blooms at a global scale. These weren't local papers, but HABs have increased on a global scale, partially due to climate change. Those data are, somewhat, people are still working on them, but that does seem to be the general trend.
Can I just add something on the modeling side? One of the things we do on the computer modeling side, so we do obviously the sensor data capture, which is the sort of ground truth data. Then we do, wherever we can, computer modeling to better understand the basic characteristics of the lake, whatever the functions are, et cetera. When we run those models, we get sort of short time frames, like 72 hours, a picture of what's going on. Very accurate because it's all validated. But the real question is what happens with climate change and things over longer periods of time. So the other alternative, which I mentioned, is that we built a model that we call the scenario engine, which allows us to do these what-if calculations at the decadal scale and above, about out to 100 years. And we developed this at Lake George, specifically first looking at understanding lake resilience. So the question actually came up of, well, one of the major issues there was salt incursion, right? Road salt incursion. And, if you shut off all the anthropogenic inputs, so if you shut off all the road salt, basically you can't do that, but theoretically, if you turn it off, what chance does the lake have to return to its state if 1983 or something, or let's say 30 years, 40 years ago, when we first started making measurements out there? And we actually came up with a number of different climate scenarios, what those possibilities are. So we could basically determine that you weren't at a point of sort of no return, that you never be able to bring the lake back. So that's one of the things we do, and we have the ability to overlay those climate models and mix them. We've actually used those to help drive programs around Lake George for salt reduction, basically going out because we had measurements to test it. So the models weren't just, yeah, it's good physics, it should work. It was, we actually tested the models against older data and basically, proposed a reduction program for certain areas of the watershed, which are now being implemented. And again, we're in a situation now where we can go ahead and track those and see how effective the programs are. So that's a really important thing. And you can put in all kinds of different variables and sort of turn the knobs and say, okay, well, what happens if this happens? What happens if the nutrients go up? What happens if nutrients go down? What happens if you clean up one stream? And then tell me how that varies for the next 20 years over different possible climate models, many of which are hotter, colder, many of which are very wet or drier. So that's another area of research that we're pursuing because it's really, really important for the long range, because all this stuff didn't happen overnight. So it's not going to be corrected overnight. We know that.
Thanks, Harry. I've got a lot of questions here, which is great. I apologize in advance. We probably won't be able to get to them all. But here's another one. Has the Jefferson Project studied the bottom sediment, I think the parentheses muck, impact on nutrient loading? Does less muck correlate to higher oxygen levels? Having a hard time reading the last bit, but basically I think you can go from there. So question about sediment and nutrients.
Okay, so we have done quite a bit of studying on this internal loading question. The fact that it's Muck, I guess, doesn't necessarily have that much to do with how much phosphorus is released as the amount of metals in the sediment, actually, because the phosphorus binds to things like iron. And if there's a lot of iron in the sediment, which we haven't exactly tested iron directly, however, there seems to be enough that phosphorus is getting bound in the sediment. So we have tested the sediment release. We've tested the amount of phosphorus in the water that gets released in these low oxygen events, and there seems to be quite a bit. We have not yet done a sediment survey. That is something we're thinking about because I think it would be, it could be very interesting and help us answer this question. So yeah, that's a really good question. It's something we're looking into. We think the sediment is a big piece of it.
And it is in plan for the next phase of the project.
And I think you basically answered the next question here, which was in your presentation, you talked about low O2 releasing the phosphorus, but if I've got it right, you could expand on it, but it has to do with that interaction with when the iron binds the phosphorus, right? So maybe just take a minute on that.
Yes, iron, and there's some other metals that can do that as well. But if there's a lot of oxygen in the system, the phosphorus is bound up to those metals, so it's staying in the sediment. And then when there's low oxygen, there are chemical reactions that happen that unbind those metals and the phosphorus, so the phosphorus gets released.
This question here, is there a trigger relationship between toxic HABs and ALS? I think there may be some research on that. I don't know how far along you are on that.
So that is not something we're directly testing. There has been a lot of research on it lately from other groups, including a group at Dartmouth that has been leading some of that research. Currently, it's very controversial, actually. There have been some correlations with blooms and increased incidences of ALS in certain areas. However, not all of the other environmental drivers outside of algae blooms have been thoroughly studied. So that's really still up in the air. There is a lot of active research in that area going on right now, though.
And I'll also mention that we've been running a little bit of side research on that, building actually some equipment. Vince Moriarty's been leading that on our side, who has been looking at a collection of aerosols. And you may have seen him on some docks, especially down Celeron area over the years, basically looking at if they could, collect airborne toxins. So we're working on that.
Okay, thanks. Next question here. You mentioned that algae growth is related to phosphate loading, yet you also noted that phosphate availability is high in the northern basin, while algae growth is higher in the southern basin. While existing algae may use up the phosphate, why aren't there more blooms in the northern basin as a result of the higher phosphate loading?
That is a very good question, and it's a very confusing situation. So one of the reasons is that the North Basin is much deeper, so there are areas of the lake where there's not enough light for algae to grow, so they can't use the entire water column in a lot of areas. Another reason is that the South Basin tends to be a bit warmer because it is shallower, and warmer temperatures tend to increase algae growth. And so it's true that nutrient is one of the main drivers of algae, but there are a lot of other drivers as well, things like lake depth and temperature and some other factors as well. But yes, that's a very good question.
There's a few questions on the relationship between aquatic plants and HABs. So I'm not sure how much you've studied that, but I'll pose one and you may get to the others because they're all related. Is the overgrowth of lakeshore aquatic vegetation increasing or decreasing the occurrence of HABs, or really how do those relate if there is a relationship?
So that's something we have not studied the macrophytes to this point. It's something we're interested in studying. So the macrophytes and the algae are both using the same nutrients. And so it's possible that in some situations they're competing for nutrients, although the macrophytes can get sediment nutrients easier than the algae can. But there is probably some interaction where there's some competition for nutrients. But yeah, we haven't studied macrophytes to this point. We do have some plans to study how the different macrophyte treatments might affect nutrients and how that might in turn affect algae blooms. But yeah, we don't have any solid answers at this point.
Okay, there's a bunch more questions. Sorry, we're not going to have time to get to them all, but thanks to Allison and Harry for being here to present, and thanks for all the great questions.
Thank you.